In this video, we're going to look at genetic exchange in bacteria, which is often referred to as horizontal or lateral gene transfer. That is, I'm going to talk about how bacteria can share DNA. Bacteria, as you know, typically increase in numbers through what is effectively cloning. That is, through making exact copies of themselves. However, in evolutionary terms, this approach has distinct limitations, as the only way that bacteria would evolve is through beneficial random mutations being passed directly on, as opposed to being shared between bacteria through sexual reproduction. Before we proceed, three key points need to be remembered. 1. Unlike eukaryotes, bacteria are haploid. 2. Their chromosomes are circular. And 3. Bacteria reproduce asexually. Hence, there are no crosses possible to explore their genetic makeup or for bacteria to spread genes through the population. So how do bacteria exchange DNA? Well, there are three ways that bacteria can share DNA. 1. Conjugation. This involves direct physical contact between the bacteria using a sex pilus. 2. Transduction. This involves viruses shuttling the DNA between the bacteria. And 3. Transformations. This is where the bacteria takes up DNA from the environment. Okay, so let's have a look at the three methods. First, conjugation. This happens when two bacteria love each other very much and decide to share some DNA. I'm kidding. In conjugation, we have a donor bacteria and a recipient bacteria. The donor bacteria contains an F plasmid on which is encoded the proteins to make the F sex pilus and the proteins needed to drive the transfer of DNA. The F plasmid can also replicate its DNA, hence it is maintained across generations of the cells. The bacteria make direct contact with each other using the sex pilus, which is a hollow tube down which the bacteria can pass single-stranded DNA to the recipient. The DNA is transferred by a mechanism called rolling circle replication. That is, the process starts at the origin of transfer in the donor plasmid, with one of the strands of DNA being passed via the sex pilus to the recipient cell. As the process occurs, the single-stranded DNA left behind in the donor cell is repaired by a DNA polymerase to become double-stranded, and likewise with the single-stranded DNA passed to the recipient cell. This process was discovered during work on auxotroph and prototroph bacteria. Auxotroph bacteria cannot make all the nutrients they need to live, and they have to pull the missing nutrients in from the environment. Prototroph bacteria can make all the nutrients they need to grow using the basic building blocks in the environment. In experiments, it was found that when the bacteria that had different nutritional requirements, that is, they required different biological complex molecules to be present in the environment, were mixed and plated out on a media lacking those molecules, the bacteria could grow. Whereas if they weren't mixed and were plated out individually, they could not. This observation suggested that there was genetic exchange occurring as the bacteria had to be acquiring the genes they needed to make the molecules that they had previously been unable to make. Here is a relatively simple example that shows this in action. Say you have two bacteria, one that cannot make serine and therefore needs to get serine from the growth media, and another bacteria that needs methionine from the growth media. And say the bacteria that cannot make serine is also able to conjugate with other bacteria and pass on genes. That is, it's a donor bacteria. If both bacteria are plated out individually on media lacking both serine and methionine, then neither will grow, as they are both unable to make one of the amino acids they need to survive. However, if the bacteria are mixed and allowed to conjugate, then when you plate out the mixture on media lacking both serine and methionine, you will get some colonies, as the donor bacteria, the one that needs serine to grow but can make methionine, has passed on the genes that can make methionine to the other bacteria that originally lacked the gene. That is, the original methionine lacking bacteria can now make all the methionine it needs thanks to the donor. Now, not all bacteria can conjugate, and not all bacteria can act as donor cells. Only bacteria that contain a small plasmid, in addition to their chromosomal DNA, can act as donor cells. This plasmid is called the F factor. The F stands for fertility. The cells that have the F factor are called F+. Cells that do not have the factor are called F-. Interestingly, the naturally occurring plasmids we find in bacteria can be divided into 5 groups. Fertility plasmids, which we have just talked about, resistance plasmids. These give antibiotic resistance and so are really bad news in the wild but dead handy in the lab. Degradative plasmids, for examples, these could be good news for the breakdown of plastics in the environment. Cold plasmids, these carry genes that kill other bacteria. And finally, virulence plasmids, which turn bacteria into pathogenic strains. Next, transduction. In transduction, a virus infects one bacteria, and during the process of making copies of the virus, it picks up some of the bacterial DNA. This DNA is then passed on to the next bacteria the virus infects. The DNA is incorporated into the genome of the newly infected bacteria by recombination. Transduction can be used in the lab to move DNA between bacteria. Finally, transformation. When a bacteria dies, it releases DNA into the environment. This DNA can then be taken up by nearby bacteria and, using recombination, incorporated into its genome. And transformation is a common technique used in the lab. Okay, so in this video we've had a quick look at how bacteria can share DNA.